that we hand them out for free. So these are open source. I made a QR code with a PDF, so people can download the file. Those are not these PDFs. This leads to the Coral Reef Arc project.
Yep.
And then this one leads to the page on this project on my website. And the idea is that this creates awareness about the role of viruses in a coral reef and also about the reef conservation project. So it's an educational and awareness thing, and the idea of using the art, the colorful object that will catch people's attention, and then we have these snippets of information. And also the information can have different levels. So this one was made for the conference, so it's not super complex, but it's still somewhat sciencey in its language. So say if it's not suitable to use, say, for high school students, I'd say, but it's more than good enough for fellow scientists. So and we repeat the information a little bit. So, and that's good, because if people only take one sheet, then so this is another one where again you have the opposite of a healthy ecosystem with lots of corals, the apex predator and lots of fish, and then the impoverished environment where there's more bacteria in the water. And there's actually a special type of bacteria they called Vibrio. They're related to cholera. So these blue ones I didn't draw these. These were photographs of the actual microscope phages, and I sort of made them into this hexagon pattern. Just for fun. And that sort of became the iconography for the viralized environment versus the microbialized environment. And if you construct this one, you get this shape constructed. And so here you can see really clearly like the healthy side and the impoverished side of the reef.
So this is the impoverished side we're looking at now?
Yes, because you see there's little fish and there is very little coral visible. And that has to do with the oxygen content in the water. So a viralized environment usually also means it's an oxygen-rich environment, which is necessary for the corals to grow and for the microbes to be down. Because one of the reasons why the oxygen content gets down in the seawater is because there's lots of microbes, they use it up. And then the oxygenation is something that the corals want to, So another idea, And the reason of this shape is because this marine biologist, has the Coral Reef Arc project. The first one that exists is a icosahedron, and that's because that they're trying to regrow lots of artificial reefs in the Caribbean, and most of them are structures that are fixed on the sea bottom, and the arc is actually a structure that's suspended from a line in the water column. And a reason to do that, the scientific theory behind it, is that if you lift the structure for the coral to grow on higher up in the water column, it's a healthier environment. On the sea bottom there's more bacteria, there's lesser oxygen, it's the pathogenic side. And if you lift it up in a water column where it's a bit, better current, better flow, more oxygen, it's more viralized, and then the theory is that the corals will grow faster on a structure in that part of the ocean than on the ocean floor. So they're doing research now. They have a few arcs out there for a year or two, and they're measuring how fast the corals are growing. And initially the rate of growth was higher on the arc. And the other clever thing about it is that it's movable, because a lot of these structures in Miami, they got cooked last summer when the water temperatures were 33 degrees or, higher, and the corals massively bleached, and lots of them died of these, artificial reefs.
The tethered ones.
And yeah, they're mostly tethered. And so the idea is too that this one you could, in theory, move to maybe a colder spot and of course there's a depth limit because the light still needs to reach it for photosynthesis.
Imagine if you had a thermostat on these and they could get themselves into a better situation.
Yeah, and the idea is too that, if these units get grown, you can also eventually sink them and then use them as building blocks for a reef. So yeah, I thought that was a very imaginative idea. So I proposed to create this floor plan idea was the theory. So for now, they only have the shape of icosahedron, and the marine biologist Forrest Rower suggested I would use these irregular shapes. These are actually existing shapes of existing viruses, and this one is not a phage, it's HIV. And but the interest that he had for the irregular shapes was that possibly an irregular shape would be really interesting also for coral arc. If you would use it as building blocks, these are great irregular shapes, but also the hydrodynamics of the water that flows around the structure changes. And so these pointy bits could, and that's why I created more corals here on these points, because I sort of theorized, I don't know how the hydrodynamics are around a structure like this, but say the water flows, more turbulently around the points, then that would be the spot where the corals would preferably start to grow out the best.
That's a beautiful representation of a theory.
Yeah. And yeah, they're kind of interesting shapes. So I have 6 models, 6 different shapes. And in these very irregular sort of bulbous ones, I did what I call the storytelling representation. And then there's a couple of elongated ones, and there I sort of just played around with the structure and the way these things are constructed, because they're all little hexagons in here behind the pattern. You can probably see it with this hexagonal shape. And in the others, I sort of made the hexagon shape very clear, but it's that hexagonal shape that's continuous that encloses actually this structure. And that's also how the virus capsid works. Each hexagon is basically one of the proteins that create the capsid shape. So there's a lot of virus experts who, research the shape of these things and then what proteins form this structure. And the proteins that enclose a virus capsid are both very strong because they need to contain their DNA, their genetic material, and at the same time they should also be kind of flexible and be able to infect a cell, enter it. So there are these, we all know about spike proteins of COVID, right? So all these virus shapes also have these proteins they need to latch on. And if it's a phage, it has this landing apparatus. So here's a phage. And so you see that the capsid is this part, which is something like irregular icosahedron. And then it has this, and sorry, I don't know the scientific name for it, this spindle and these sort of spindly legs. So it can actually, you can see here, it lands on a bacterium, and then it sort of empties out its DNA into a bacterium, multiplies, and then it bursts open. And then in some instances, it won't destroy the microbe, and then the DNA will remain inside, and the bacteria will multiply with the embedded viral DNA or RNA. There's different types I've learned. And then they become actually, here they destroy all the bad bacteria this way. So they act as a external immune system in a way. They keep the corals healthy because they live in the mucus. So this is a coral polyp, and this blows up, so you can see all the little phages happily living in the mucus layer of the coral polyp. And they go out and they attack all the microbes that are unwanted, and they destroy them. So they keep the corals healthy. So they're actually really good for the reef. But then if the environment deteriorates, the virus changes tactic, then it's more advantageous for it to not destroy the microbe, but inject their DNA or RNA and keep the bacterium living. They call that piggyback the winner, because then the microbes in this environment are the winners. They're multiplying, they're thriving on this, soup of algae and photosynthesis, dissolved organic carbon. They love it and they multiply. That also means they deplete the oxygen in the water. And that's all very disastrous for a healthy reef system. And then you get this flip to a green water system with algae and no corals. Less fish, less species. That's the story.